Hello, my name is Michael Hilliker, and this is my reproductive system portfolio. So I did my presentation on the determination of sex and the roles of genes versus hormones in guiding the development of the reproductive system and just the system and the body as a whole. So I felt it was important to start off with a little bit of background information. So to begin, the human genome programs sexual development. And this is important because we have 23 pairs of chromosomes, or 46 chromosomes overall, that will guide that development within us. So the genetic sex of an individual will depend on the sex chromosomes, female being XX and males being XY. So in the absence of a Y chromosome, an embryo will develop into a female. Now the male and female sex organs both consist of gonads, internal genitalia, and external genitalia, and these will differ once their development occurs. So in a broad sense, internal genitalia includes all of the accessory glands and ducts that connect the gonads with the outside environment. External genitalia would include all of the external reproductive structures that are visible in the external. And males and females both do have gonads. So males have testes and females have ovaries. So now getting into more of the genes and hormones that will guide the determination of gender within. So the determination of sex will depend on the presence or absence of the sex-determining region of the Y chromosome. So here is a picture over here showing the Y chromosome and pointing right here, the sex-determining region so the SRY gene is actually right there. If the SRY gene is present and functional, the bipotential gonads will develop into a testes or into testes. So it will take on more of the male characteristics and become a male. If the SRY gene is absent, the gonads will develop into ovaries, and this will be under the direction and guidance of multiple female-specific genes. Now moving on to the male embryonic development. So once the SRY gene is present and becoming a male, the SRY gene will produce a protein that is called the testes determining factor, or TDF for short. So TDF will bind to DNA, and this will activate additional genes that include the SOZ sub 9, WT sub 1, and SF sub 1. So the protein products of these and other genes will direct the development of the gonadal medulla into a testes. Now it's also important to note that testicular development does not require male sex hormones, such as testosterone. The developing embryo is actually unable to secrete testosterone until after the gonads differentiate into testes. Now there are 3 hormones that are secreted from the testes once they differentiate. So these are the Sertoli cells, and these secrete a glycoprotein anti-Müllerian hormone, or AMH for short. Testicular interstitial cells, or Leydig cells, as they're also known by, secrete the androgens testosterone and the derivative of testosterone being dihydrotestosterone, or DHT. Moving on to female embryonic development. So if the SRY gene is absent, this will occur. The cortex of the bipotential gonad will develop into ovarian tissue, and multiple genes are required for the development of functional ovaries. Now, in the absence of testicular AMH, or anti-Müllerian hormone, the Müllerian ducts will develop into the upper portion of the vagina, the uterus, and the fallopian tubes. Now, when testosterone is absent, the Wolffian ducts will degenerate. So they will no longer be there. And without dihydrotestosterone, the external genitalia will take on female characteristics as well. Now, these last two slides are more of a summary of when the different types of genes and hormones will determine what develops and what doesn't develop if they're present in absence. So more of a couple of summary slides, but important to note just when they occur and how they occur. So at the 6-week mark, if it is a female, the gonadal cortex will form the ovaries, the gonadal medulla will regress, the Wolffian duct will regress because testosterone is absent. The Mullerian duct will become the fallopian tube, uterus, cervix, and the upper half of the vagina. And at this point, anti-Mullerian hormone is absent. At 6 weeks, if it is a male, the gonadal cortex will regress, the gonadal medulla will form a testes, Wolffian duct forms the epididymis, vas deferens, and seminal vesicle with the testosterone being present. So again, taking on the male characteristics. And the Mullerian duct will regress because AMH is present. Now at 10 weeks, if it's a female, the gonadal cortex will become the ovary in the absence of the SRY protein and under the influence of female-specific genes. The absence of testosterone causes the Wolffian duct to degenerate. At 10 weeks, if it's a male, the SRY protein in the male embryo, again, will direct the medulla of the bipotential gonad to develop into testes. The anti-Mullerian hormone from the testes causes the Mullerian ducts to disappear. And everything that would be present at birth because of all of the development throughout those last two cycles at the 6-week and 10-week mark. The female will have the absence of anti-Mullerian hormone because that allows the Mullerian duct to become the fallopian tube, uterus, that part of the vagina. At birth, for the male, it'd be male at that point because the testosterone from the testes converted the Wolffian duct into the seminal vesicle, again, the vas deferens, and the epididymis, because dihydrotestosterone controls the prostate development. DHT also causes the development of the male external genitalia, and in the absence of androgens, the testosterone and dihydrotestosterone, the female external genitalia are produced. Now, the picture, the little diagram off to the side here, I thought it was a nice representation of seeing visually how everything develops in the male and female. So from the bipotential gonads, if there are male hormones, so the SRY gene and the SRY proteins, you'll see the epididymis, the testes, vas deferens, and the seminal vesicle. And without the male hormones on the female side, the female gonads will turn into the ovaries, also the oviduct, uterus, and the cervix. And now, moving on to the study guide, or the reproductive system worksheet that I created. So I made it with matching terms. So being that the terms can be used once, twice, or not at all, I thought it was a nice way to learn the material because there are a lot of genes and hormones that act to develop male and female characteristics. So the number of chromosomes in the human genome would be A46. The SRY gene turns the bipotential gonads into testes. The Sertoli cells secrete anti-Mullerian hormone. The Leydig cells secrete testosterone and dihydrotestosterone. The absence of DHT causes the external genitalia to take on female characteristics. Testosterone being absent causes the Wolffian ducts to degenerate. And the absence of AMH causes Mullerian ducts to develop into the upper portion of the vagina, uterus, and fallopian tubes. DHT promotes the development of the prostate gland and external genitalia, and the SRY gene being absent causes the gonads to develop into ovaries. And that will end the consideration for the male and female development because of the genes and hormones. So I thought it was nice to see just how the presence of one, the absence of one, it really all connects and just shows the development as a whole.